Chapter 2 through 4, Cellular Homeostasis and the Structure and Function of Hemopoietic Organs. When we talk about tissue homeostasis in hematology, it refers to the maintenance of an adequate number of cells to carry out the organism's functions. So if we're talking about a human body, or somatic cells, including the blood cells, they have to go under certain fates. So those fates include proliferation, and this happens by mitotic cell division. They have to differentiate and acquire specific cells for specialized functions, and they have to die and be eliminated from the body. So cell proliferation is required for the replacement of cells lost to terminal differentiation, cell death, or cell loss. Differentiation provides a variety of different cells, each of which is capable of executing specific and specialized functions. Cell death is an active process that can self-initiate. Apoptosis is physiologically important as cell proliferation and differentiation in controlling the overall homeostasis of various tissues. When we talk about a cell in hematology, we are going to consider it in three different parts. First, the plasma membrane, then the nucleus and the cytoplasm. So let's discuss the plasma membrane. It serves as a semi-permeable outer boundary separating the cellular components from their surrounding environment. And it serves for the following basic functions. It provides a physical and flexible barrier to contain and protect all of the cell components from the extracellular environment. It regulates and facilitates the interchange of substances with the environment by endocytosis, exocytosis, and selective permeability. It establishes electrochemical gradients between the interior and the exterior of the cell, and it has receptors that allow the cell to respond to a multitude of signaling molecules through signal transduction pathways. Now, the nucleus is composed of three components: the chromatin, the nuclear envelope, and the nucleoli. It is basically the control center of the cell and the largest organelle of the cell. The nucleus is composed largely of DNA and is a site of DNA replication and transcription. It's responsible for the chemical reactions within the cell and the cell's reproductive processes. The nucleus has an affinity for basic dyes because of the nucleic acid contained within it. It stains a deep purple with right stain. The cytoplasm matrix is homogeneous, continuous, and aqueous. It is the environment in which all of the organelles exist and function. The organelles within the cytoplasm include the ribosomes, which synthesize proteins, the rough ER, which synthesizes most membrane-bound proteins, the Golgi apparatus, which modifies and packages all of the macromolecules for other organelles and for secretion, the mitochondria, which produces most of the cell's ATP by oxidative phosphorylation, lysosomes, which contain enzymes that degrade any unwanted material. Now we'll discuss the cell cycle. The purpose of the cell cycle is to replicate DNA once and distribute identical chromosome copies equally to two daughter cells during mitosis. The cell cycle is a biochemical and morphologic four-stage process through which a cell passes when it is stimulated to divide. The stages are G1 or GAP1 phase, S, which is a DNA synthesis phase, G2, which is a GAP2 phase, and M phase, which is mitosis or cell division. When we look at the cell cycle, we begin at G1 phase. So it's a period of cell growth and synthesis of components that are necessary for replication. In the S phase, DNA replication takes place. An exact copy of each chromosome is produced and they pair together as sister chromatids. The centrosome is also duplicated in the S phase. When we get to the G2 phase, the tetraploid DNA is checked for proper replication and any damage. If it's okay to proceed, the G2 will go into the M phase, and mitosis and cell division will occur. Now, at G0, or quiescent stage, the cell is not actively in the cell cycle, so it could just be waiting there for stimulus. A regulatory mechanism is needed to prevent abnormal or mutated cells from going through the cell cycle and producing an abnormal clone. It's a very highly complicated process. So there are different checkpoints that need to be initiated. The first checkpoint is at the G1 phase, and this is checking for appropriate amount of nutrients and the appropriate cell volume to proceed. The second checkpoint is at the end of the G1 phase into the S phase checkpoint. This checks for DNA damage and makes the cell wait for any DNA repair, or it will initiate apoptosis or cell death. The next checkpoint is in the G2 DNA damage checkpoint phase. This takes place after DNA synthesis and at the end of G2, and its purpose is to verify that replication took place without any error or damage. If there's any abnormal or malformed replication that has occurred, mitosis will be blocked. Now the last checkpoint is during mitosis at the time of metaphase. Here, the attachment and alignment of chromosomes on the mitotic spindle and the integrity of the spindle apparatus are checked. If there's any abnormalities, anaphase will be blocked. So let's look at the cell again and see what fates are possible. So quiescence is the waiting phase. This is the G0 phase. Once signals to replicate are given, the cell enters into the cell cycle. Now here, they can wait for more signaling, called the senescence phase. They can terminally differentiate into a specific cell, for example, neutrophil. If there's any error or unchecked regulation, there will be neoplastic transformation, causing cancer cells. Or those errors can be tagged and the cell could be programmed to die. If all goes right and all the checkpoints are greenlit, the cell will go into proliferation to make new cells. Let's check a system. If there are errors that occur and it is tagged for program cell death, we are entering into the ubiquitin proteasome system. So any molecules that are destined for destruction, they're tagged with a small amino acid polypeptide called ubiquitin. Those appropriately labeled molecules are then transferred to ATP-dependent protease complexes for destruction. Generally, these proteins bearing a single molecule are marked for endocytosis and degradation by those lysosomes within the cytoplasm of the cell. Once they're checked multiple times and there are multiple errors, they are marked for destruction and assembled into a cylinder, and the channels which the proteins are given set it up for destruction. If we look at hematological cells, they all begin with the hematopoietic stem cell. Differentiation is a process that generates the diverse cell populations found throughout the body. All cells contain the same genetic information. The appearance of specific characteristics in various cell populations is dictated by specific genes that are actively transcribed into messenger RNA and the translation of that genetic information into functional proteins. Now, the regulation of this gene expression is controlled at various levels. For differentiation to occur, there must be regulation of certain processes. So, for example, a cell can differentiate by caused epigenetics. This is a stable change in the gene function that silences certain genes so cells can become one specific cell. Another way this can happen is through translational messenger RNA. So there's two forms of RNA involved in regulating the translation of cells. Both molecules function in a pathway and block specific protein expression by inhibiting translation or inducing degradation of their respective target, which also silences certain genes so cells can become one given thing. Two major mechanisms for cell death: necrosis and apoptosis. Necrosis is a pathological process caused by direct external injury to the cells. For an example, a burn, radiation, or toxins. Apoptosis is a self-inflicted cell death originating from the activation signals within the cell itself. Most apoptosis occurs as a normal process to eliminate potentially harmful cells, or cells that are no longer needed, or aging cells. So apoptosis is considered cell suicide, where necrosis is considered cell murder. Apoptosis of older, terminally differentiated cells balances with new cell growth to maintain the needed numbers of functional cells in the organs, hematologic tissue, and epithelial cell barriers. On the other hand, apoptosis can also initiate in response to internal pathological injury, like we saw with the last ubiquitin system when there was DNA damage during the cell cycle. Let's look at Table 2.3, which will help you determine the differences in the way cells die. So either through necrosis or apoptosis. So usually the result for necrosis is an external stimulus, like toxins or massive insult or injury, where apoptosis is usually a physiological condition that doesn't need to occur with energy. As the cells break down, in necrosis, the cells are broken. The cell membrane is broken and the cell is lysed. In apoptosis, the cells remain intact and they are blebbed with alterations, and you start to see the loss of phospholipid asymmetry. Now we've spoken about the balance of cells, let's talk about hematopoiesis and the functional organs in hematology. So hematopoiesis is a continuous, regulated process of blood cell production that includes the renewal, proliferation, and differentiation, as well as maturation of cells. It begins in humans by a selected distribution of embryonic cells in specific sites that rapidly change during development. So in healthy adults, hematopoiesis is restricted primarily to the bone marrow. But during fetal development, as you can see in this chart, its restricted distribution cells initiates in the yolk sac and then progresses in the aorta, gonad, mesonephrons region, then into the fetal liver or the hepatic stage, and finally residing in the bone marrow or the medullary stage after about 6 months gestation. Now, due to the different locations of the microenvironmental conditions, Each of these locations has a distinct but related population of cells. In adults, hematopoietic tissue is located in the bone marrow, lymph nodes, spleen, liver, and thymus. The bone marrow contains developing erythroid, myeloid, megakaryocytic, and lymphoid cells. Lymphoid development occurs in primary and secondary lymphoid tissue. Primary lymphoid tissue consists of bone marrow and thymus, and that's where the T and B cell lymphocytes are derived. Secondary lymphoid tissue is where lymphoid cells respond to foreign antigens consisting of the spleen, lymphoids, lymph nodes, and other mucosal associated lymph tissue. Bone marrow is one of the largest organs in the body, and its tissue location is within the cavities of the cortical bones. Normal bone marrow contains two major components: the red marrow, which is hematologically active marrow consisting of the developing blood cells and their progenitors, such as myeloid cells, erythroid cells, megakaryocytic cells, in very early stages of the lymphoid cells. And the yellow marrow, where there's hematopoietic inactivity because it's restly or mostly composed of fat cells. Now, the thymus, spleen, and lymph nodes are sites of later lymphoid cell development and are considered into primary and secondary lymphoid tissues. Within the bone marrow environment or microenvironment, there are specific niches or little islands where specific cells are made. They play an important role in nurturing and protecting the stem cells and regulating a balance among initiating the quiet phase, the self-renewal phase, and differential phase. In order to maintain all of these niches, the marrow has a good circulatory system. The nutrient and oxygen requirements of the marrow are supplied by the periosteal arteries, which are near the bone foramina. The nutrient artery supplies blood only to the marrow. It coils around the longitudinal vein, which passes along the bone canal. In the marrow cavity, the nutrient artery divides into ascending and descending branches that coil around the longitudinal vein. The arterioles branch and enter the inner lining of the bone, which connect the capillaries and provides a rich nutrient flow. Here you can see a cross-section of a bone where you see the yellow marrow composed of fat and the red marrow where the cellular niches will be. Then you see the expansive vasculature network within the bone marrow to supply enough oxygen and nutrients to those developing cells. Once they mature, they egress or come out of the bone marrow to go into peripheral circulation. Any cell made within the bone marrow is called medullary hematopoiesis. If there are hematological cells made outside of the bone marrow in organs such as the liver and spleen, this is called extramedullary hematopoiesis. At birth, the thymus is an efficient, well-developed organ and consists of two lobes measuring about 0.5 to 2 centimeters in diameter and is further divided into lobules. The thymus is located in the upper part of the anterior mediastinum at about the level of the great vessels of the heart. It resembles other lymphoid tissue in that the lobules are subdivided into two areas. And on the right, you see a cross-section of the thymus, where you have the cortex and the medulla, which is in the middle. The cortex is on the outside. Both areas are populated with the same cellular elements: lymphoid cells, reticular cells, epithelial cells, and macrophages. The main function of the thymus is to get signals of immature T cells and mature any T cells within thymus itself. The spleen is the largest lymphoid organ in the body. It's located directly beneath the diaphragm, behind the fundus of the stomach, in the upper left quadrant of the abdomen. It is vital but not essential to life and functions. As it does filter circulating blood, the spleen in a healthy individual contains about 350 milliliters of blood. The exterior surface of the spleen is surrounded by a layer of peritoneum covered by connective tissue capsule, and you can see that in the right side cross-section. The capsule projects inward, forming tuberculae that divide the spleen into discrete regions. Located within the regions are three types of splenic tissue: white pulp, red pulp, and a marginal zone. The white pulp consists of follicles and germinal centers containing lymphocytes, and macrophages. Aggregates of T lymphocytes surround the arteries that pass through these germinal centers, forming a region called the periarterial lymphatic sheath. And here there is mainly B cells. It has a very special role. It has two pathways: a quick transit pathway that filters blood throughout the body and provides oxygen to the tissue, but it also has a slow transit pathway that plays a very important role in red cell sinistering. So first, it goes slowly through its vasculature, and it pulls the red cells in to filter it and check for any defects so they can destroy it. If there are any defects or inclusions, it will pit out or pluck out any particles from the red blood cells. It also provides immune defense in rich phagocytic cells and lymphocytes. And in addition, it stores a lot of platelets. Lymph nodes are organs of the lymphatic system located along the capillaries that are parallel but not part of the circulatory system. The nodes are bean-shaped structures, either 1 to 5 millimeters in diameter, that occur in groups or chains at various intervals along the lymphatic vessels. They may be superficial, like within the armpit, or they can be deep in the mesoteneric retroperitoneal system. Lymph is a fluid portion of blood that escapes into the connective tissue and characterized by low protein concentration in the absence of red cells. On the right cross-section of a lymph node, it can be divided into an outer region called the cortex and an inner region called the medulla. The outer cortex contains tuberculae that provide support for macrophages and lymphocytes located within the node. After antigenic stimulation, the cortical regions of some follicles develop activated B cell proliferation and are called germinal centers. Follicles with germinal centers are called secondary follicles, while those without are called primary. Mucosal-associated lymphoid tissue are collections of loosely organized aggregates of lymphocytes found throughout the body in association with mucosal surfaces. Its basic organization is similar to a lymph node in that T and B cell rich areas can be identified but not clearly demarcated as in the lymph nodes. The medulla is not present as a separate structure, and no capsule can be identified. In the intestine, some of these aggregates are known as Peyer's patches. Tonsils and the appendix are also part of the malt. Its function is to trap antigens that are crossing mucosal surfaces and initiate immune responses rapidly. Now we're going to talk about the concepts of cellular commitment and differentiation in the hemopoietic system. It discusses the characteristics that define the precursor cells and the cytokines that regulate the development of these cells. The structure and function of the receptors are presented with their signaling pathways And microenvironments also need to be described so we can understand fully how cells are made in hematology. Differentiation is responsible for generating the diverse populations that provide the specialized functions needed by the human. Differentiation is defined as the appearance of different properties in cells that were initially equivalent. Because all cells of a human carry the same genes, differentiation, or the appearance of specific characteristics, occur by the progressive restriction of other potential developmental programs of the cell. So commitment is defined as the instance when two cells derived from the same precursor take a separate route of development. It assigns the program, and then maturation begins. The bone marrow is estimated to be capable of producing approximately 2.5 billion red cells, 2.5 billion platelets, and 1 billion white cells per kilogram of body weight daily. Now, hematopoietic precursor cells can be divided into 3 cellular compartments defined by their relative maturity. They are the hematopoietic stem cells, hematopoietic progenitor cells, and maturing cells. So from the hematopoietic stem cell, we have signals to commit to a specific progenitor cell, and then we differentiate into maturing cells. Hematopoiesis is derived from a pool of undifferentiated cells called hematopoietic stem cells. They give rise to all bone marrow cells by the process of proliferation and differentiation. Now, the compartment within the bone marrow microenvironment is the smallest. They only constitute about 0.5% of the total marrow nucleated cells. These are very special cells. They're rare, but they are capable of regenerating the entire hematopoietic system. They're defined as multi-potential precursors because they can maintain the capacity to give rise to all lineages of cells. The other defining characteristics is their high self-renewal capacity. They can give rise to daughter stem cells that are exact replicas of the parent cells. Stem cells have 4 possible fates. Just like we saw with the cell cycle, cells can be in the quiescent phase, the G0 phase, waiting for stimulus to commit to a certain cell line. One thing that a stem cell can do that no other cell can do is they can self-renew. They can also commit or differentiate to a maturing cell, or they can be programmed for cell death if any errors are detected. To meet all the demands imposed on the hematopoietic system, some of the stem cells from the stem cell compartment initiate differentiation. The transition from the stem cell to a committed progenitor correlates with the downregulation of stem cell-associated genes via gene silencing and the upregulation or activation of lineage-specific genes. And after additional divisions, the daughter cell progressively loses their ability to generate cells of multiple lineages and gradually becomes restricted in differentiation to a single cell lineage. The compartment also includes precursor cells developed and located between the stem cells and the morphologically recognizable precursor cells. After a series of amplifying cell divisions, the committed precursor undergoes a further change when the cell takes on the morphological characteristics of one cell lineage. Maturing cells constitute the majority of the precursor cells. proliferation, and amplification boost the cells to 95% of the total precursor cell pool. The capacity to proliferate is lost before full maturation of these cells are complete. They exhibit the recognizable nuclear and cytoplasmic morphological characteristics that can be used to classify their lineage and stage development, as you saw with the maturing cells of a neutrophil, red cell, platelet, and the other white cells. Now here you can see in figure 4.4, we have a representation of where the hematopoietic stem cell begins. With many transcription and positive cytokine factors, they get signals to stimulate to commit to a precursor and then differentiate into one of the maturing cells that we have discussed before. The regulation of the stem cell and progenitor differentiation and expansion is critical because it's determined the concentration of different cell lineages in the bone marrow and eventually into the peripheral blood. Now, specific glycoproteins called growth factors or cytokines govern the precursor cell survival, self-renewal, proliferation, and differentiation. Growth factor control of hematopoiesis is highly efficient, and there's a communication system that allows the coordinated increases in the production and functional activity of the appropriate cell type without the expansion of irrelevant ones. The first identified growth factors were described as colony-stimulating factors because they supported the growth of these colonies in vitro cultures. Now, the mechanism of cytokine regulation is often referred to a communication network where it exhibits signals in amplification circuits, including autocrine, paracrine, and juxtacrine, different mechanisms for stimulation and amplification. Autocrine signals are produced by and act on the same cells. Paracrine signals are produced by one cell and act on the adjacent cell, typically over short distances, and juxtacrine signals represent a specialized type of paracrine signaling in which a cytokine is not secreted by the cell, but produced it because it remains membrane bound, necessitating direct producer cell target cell contact to achieve the desired effect. In addition to getting the positive cytokine signal, a cell's phenotype and function are determined by the genes expressed by that cell. So differentiation is regulated by different gene expression patterns. The growth factor maintains hematopoiesis are not thought to be instructive for the pathway of differentiation, but permissive for the cell to be viable and proliferate. The components that actually establish the pattern of gene expression are considered transcription factors. These decisions are controlled by the integrated effects of signaling pathways initiated by external cytokines and internal transcription factors. Now, transcription factors are DNA-binding proteins that interact with the regulatory promoter regions of the target genes. So erythropoiesis, or the production of red blood cells, comes from its progenitor that gives rise to a burst-forming unit specific to erythrocytes and a colony-forming unit specific to erythrocytes that depend on the hormone erythropoietin, or EPO. Now, this gives rise to the first recognizable maturing red blood cell. Granulocytes and monocytes come from the same progenitor. They're derived from the granulocyte-monocyte progenitor, which also is derived from the colony-forming unit, granulocyte and monocyte. They have specific growth factors that help support their differentiation. As you can see, granulocytes have a specific designation, and so does monocytes. Megakaryocytes or thrombopoiesis, the making of platelets, are derived from megakaryocytes and come from the precursor of the megakaryocyte erythrocyte progenitor. They have colony-forming units specific to platelet production and growth factors that influence the proliferation and differentiation. They are very dependent on the hormone thrombopoietin to induce the greatest increase in platelets. Lymphopoiesis occurs in the multiple locations within the body, the bone marrow, the thymus, the lymph node, and the spleen. There are multiple growth factors that act together that play a role to help produce lymphocytes.